Hello again, this is Dr. Kevin Connors. That's my little mug down in the left-hand corner of that slide. We're going to be talking about apoptosis. So if you follow me through this series so far, this is apoptosis 301, the third in the series of our little short classes on the mechanisms of apoptosis. Now, apoptosis is normal programmed cell death. And we went over the difference between apoptosis, normal cell death of a cell, and necrosis, which is basically a cell being murdered. So this is apoptosis is what should take place. It is not taking place with a cancer patient. So if you have cancer, those cancer cells are not going through normal apoptosis. That is step one in how to get cancer. Don't have your cells go through apoptosis, and it typically starts with just one singular cell. So one cell doesn't go through apoptosis. That's not cancer in itself, but that's the first step. The second step is it goes into a rapid replication phase. So cancer is a cell that doesn't go through apoptosis. It is in rapid replication. Now it's not one singular cell anymore. It becomes a multiple of cells, and by the time it's diagnosed, it's millions of cells. So those cells replicate cells that don't go through apoptosis. So if you want to cure cancer, you have to figure out the mechanism of why that cancer cell, now cells, are not going through apoptosis. So learning this isn't just cold, dry physiology. It's cool, interesting stuff to really help you develop a strategy to deal with a growing cancer. This is what chemotherapy is trying to do. This is what radiation is trying to do. It's trying to create apoptosis through necrosis, but we want to be able to turn on apoptosis. So you understand why we're learning this stuff. If you have further questions, make sure on the upper left-hand corner of that slide it says connorsclinic.com. That's our website. There's a bunch of information on my website there too. And feel free to download my book. You can find it on the website. You can find it on the button on the slide. So just make sure you download my whole book, Stop Fighting Cancer. That will help you with this whole thing too. And you'll get on my newsletter list, and you can get all this fun stuff and learn all this stuff on how to take care of yourself. So the mechanisms of apoptosis. We talked about there's an extrinsic pathway and an intrinsic pathway leading to a final common pathway. So right now we're going to be talking about, in this presentation, the extrinsic pathway. The extrinsic pathway starts extrinsically, mean extrinsic to the cell, exterior to the cell itself, on the cell membrane. So this is a picture of the cell here. The cell is the rounded part that's gray in this picture, and this purplish double thing on this picture is a receptor on the cell membrane. This is a Fas ligand receptor, or what's called a death receptor. So the extrinsic pathway is receptor-based, meaning there's things from the outside that turn on the process of apoptosis from external to the cell. So we're going to go through this really quick. So if you really want to study this, then you got to listen to it a few times and dig in a little bit deeper. But these Fas receptors, they're tumor necrosis factor receptors, will be stimulated by a ligand. A ligand is a thing that turns on a receptor. It's kind of like a key that fits into the keyhole. The keyhole is the receptor, if you want to think of it that way. So this Fas receptors, tumor necrosis factor receptors, will be turned on. And when they are, these receptors actually migrate together, kind of accumulate together. They are proteins, and they stimulate enzymes within the cell. And this process is now turned on. The process that's turned on in this system is called the caspase system. So it activates this caspase system that is going to lead to cellular apoptosis. So the death receptor turns on, the ligand stimulates it, turns on this receptors, the receptors accumulate together, turns on this enzyme system called the caspase system. First, the procaspases are stimulated. They activate these initiator caspases, which activate the executioner caspases. And the executioner caspases are so named because it turns on a bunch of enzyme systems that start the cell to destroy itself from the inside out. This is totally an inside job now from this point on. So once the receptor is stimulated, it turns on this process so the cell will start destroying itself from the inside out. The executioner caspase starts to initiate DNA ACEs. So whenever you see ACE at the end of any word, it is really an enzyme that will start to devour or eat something up. The DNA ACEs actually start to devour the nucleus where the DNA is in the cell. So it starts to devour that so it can't reproduce itself again. That's part of the nucleus's job is in reproduction. So in a cancer patient, the cell isn't going through apoptosis, and then there's also a nuclear issue, which is why cancer is often said to be genetic, because it has that genetic component, because that nuclear reproduction of the DNA is happening very rapidly. Instead of about every 12 to 16 weeks, now it's reproducing 5 to 10 times a day. That's what cancer is. But if the apoptotic system is activated, this caspases, the executioner caspases, will activate DNA ACEs, which will gobble that up and start to destroy that. Remember, most of the nuclear components, most of the cellular components are going to be preserved. They're going to be conserved in apoptosis. So it is really like a recycling of the cell nutrients. So it's being gobbled up by these enzymes that then all the nuclear material and the cellular material starts to fragment and starts to get broken down. It causes these, here's a perfect picture of these little membrane blebs, they're called. So instead of being a circular cell, it starts to look, the cell membrane starts to look kind of weird as it starts to break apart and basically break into smaller pieces so your white blood cells can chew them up. That's what's called phagocytosis. Your own body's white blood cells, typically macrophages in the white blood cell system, will be attracted to these things and start chewing them up. Matter of fact, the surfaces of these blebs actually secrete a little protein like, you can think of it like a honey that attracts the macrophages to it, so they go, ooh, this tastes yummy, and they go gobble up these cells. Now this is taking place so many times in a given hour in your body in a normal process. Apoptosis is taking place in different cells, and your macrophages are gobbling them up and recycling the material and feeding off of it, and it's the way your body takes care of itself and recycles itself. So the macrophages are attracted to those surfaces, and they start to gobble up those apoptotic bodies. So the apoptotic bodies are the pieces of the cells that are breaking off of those blebs. This is the extrinsic pathway. So the extrinsic pathway starts with a receptor. The receptor is stimulated by a ligand. Remember, vitamin D is a ligand in the apoptosis process, so it's really important. As well as some other nutrients are really important for apoptosis. Follows through the final common pathway, what goes on inside the cell, a whole bunch of different processes, mainly through the caspase system, that then gobble up the DNA, gobble up the cytoplasm, that's the inner gel of the cell, starts to gobble up the other bodies, the ribosomes, and other cellular bodies, and the cell membrane starts to bleb out, and the macrophages gobble it up. So trying to fly through these slides, so if you missed that, go back and listen to it. But stay tuned for our next presentation on 401 when we get into the intrinsic pathway. Hopefully this was helpful. Visit our website. Call our office if you have any questions. We're always here for you. Thanks. Have a great day.